In this session, we're going to talk about RNA translation. Okay, so what are just some of the basics that you're supposed to understand? Now, in previous sessions, we talked about what we call the central dogma of molecular biology. And from the central dogma, we said your DNA is going to be, replicated to DNA. What we mean is that DNA is going to make a copy of itself. Then this DNA will be transcribed. Okay, it's going to be transcribed to RNA, but specifically to mRNA, messenger RNA. Then, since this messenger RNA has gotten the information, it's now going to be translated to proteins. Okay, so to just put it into context, DNA to DNA, we're saying this is DNA replication, DNA making a copy of itself. Then from DNA to mRNA or RNA, this is called transcription. Okay, then after transcription, now your mRNA now is going to be converted to proteins, a process we call translation. Okay, so we've talked about these two. You can watch our videos, you're going to understand. But now we're talking about translation. We want to now finish with the central dogma of molecular biology by talking about the last component. Okay, so what is translation? So when we're talking about translation, we're just talking about the making of proteins. Okay, you making proteins. Okay, a basic scenario that we gave was you're just from doing your haircut, maybe after 2 weeks or maybe the third week, hair grows. But how does it grow? So it will grow because there's a demand for it to grow. The other example that we can give is your nails. Okay, you've cut your nails today. Let's say 2 weeks or 3 weeks from now, your nails have grown back. What has happened? Okay, nails, these are proteins. Hair, these are proteins. So they need to be made. Now DNA is going to, the DNA that is specifically made for hair, it has to code for that, okay, to make those proteins. So basically that's the basic gist of it. Okay, so that is it. We are making proteins. Now, which organelles in the body, or let me say in a cell, a component of a cell which makes proteins, that they are ribosomes. Okay, ribosomes are involved in the making of proteins. Now, in the making of proteins, there are 3 things that you're supposed to understand. Okay, number 1 is the same ribosome. You have to know it in and out. Okay, structures, its units, that is very important. Number 2, RNA itself. Okay, now we've talked about RNA, nucleic acids. Basically, you can also revisit that just to get a basic concept of RNA and DNA. Okay, these are nucleic acids. Okay, and then you also have to know amino acids. Amino acids are what makes proteins. Okay, so basically the ribosomes that makes proteins. Okay, you need an RNA. Basically in this case we're talking about what? Okay, it can be the mRNA which has that information. Okay, and the ribosome RNA. These are going to play a very key role in this process. Okay, so let's talk about each of the components one by one. Let's start with the ribosomes. Okay, so the ribosome itself is got 2 units. Okay, it's got the larger subunit, or let me start with the small subunit, and the other unit is called the large subunit. Okay, so now with these 2 subunits, let me just come up with maybe a diagram to explain them better. So let's say this is a small subunit, this is a large subunit, then let's combine them. Okay, so basically this is what we have: the large, this and the combined, and the small. Okay, now that there are different types of cells. We've been talking about cells, but basically we've talked about what we call eukaryotic cells. Now eukaryote, a karyote is just a nucleus. So when you're talking about you're talking about something that is true. So these ones have got a true nucleus. On the other hand, we've got prokaryotes. They don't have a well-defined nucleus. Okay, so it's not membrane bound. So basically these are the two types of cells that we're going to talk about. However, in this whole segment of molecular biology, specifically the central dogma, we've been emphasizing that we are using prokaryotes, and the premise is that prokaryotes themselves, it's easy to study this process in prokaryotes. Coming to eukaryotes is a very complex concept. Okay, to even just talk about how RNA is being converted to proteins, that is a very, very complex process. Basically we just use prokaryotes because it's easy to understand. Now in eukaryotes, the smaller subunit, it starts at 40S. Okay, now I'm going to explain what S means in this case. Then from 40S you're going to add a 20. You're just going to be adding a 20 throughout. Okay, so for a large subunit, when you add a 20, it's going to be what? 60S. Okay, now when I combine 40 and 60, that's not the way we combine like the usual arithmetic. So like I've said, I'm just adding 20 throughout. So to 60 I'm also going to add a 20. That will just be 80S. Okay, so when I combine 40 and 60, logically should give me 100. However, we don't count it like that because I'll explain why that is the case. Then since eukaryotes are larger than prokaryotes, because prokaryotes are simple things like bacteria, eukaryotes are bigger cells like the animal cell, the plant cell. Okay, basically that since prokaryotes are smaller than the eukaryotic cells, so it means even the small subunit will be a bit smaller. So in this case, from 40, let's remove a 10. Okay, that will just give me a 30. So this will be a 30S. Okay, now the difference will just be, for this one, for prokaryotes, the difference will also be 20. So I'm just going to be adding 20. So to 30, when I add the 20, I'm going to have 50S for the large unit. Then to 50 again, I also add a 20. I'm just going to have a 70S. Okay, that is a combined 30 and 50. Mathematically, when I add 30 and 50, that it gives me 80, but that's not how we add the 70S. Okay, now this is very, very important for us to understand because it's very much examinable. Okay, and you wouldn't want to miss that. Now, what is the S? The S is what we call, it's called the Sudbeg. Okay, so it depends with what literature you're using. It's called the Sudbeg unit or whatever you might want to call it, but it's called the Sudbeg unit. Okay, others will call it a unit, others will call it a measure. But now the Sudbeg unit is just a measure of the rate at which sedimentation is taking place. Okay, the rate at which a certain unit sediments. Okay, so S in this case is representing the sedimentation. Okay, so now what is sedimentation? Not really. When we're talking about especially these units in ribosomes, the sedimentation, because sedimentation, let's say I've put something in a centrifuge, then I let the centrifuge spin at a very high, let me say, a very high rotation rate. Then I let it settle. As it is settling there, you find that in case there's something, example for, let me say, the way blood coagulates. Okay, the way blood coagulates when it's spin in a centrifuge, then within a short period of time the blood cells are going to come together and settle in one area. That is, some form of sedimentation. So basically that's what we're talking about. Okay, so that's, those are the subunits that you have to understand on the ribosomes. Then we said another thing that you need to have for you to make proteins is the RNA. Okay, now what about the RNA? You know that there are 3 types of RNAs. Okay, now we've been talking about these ones, but just to remind us ourselves. Okay, there's what we call the mRNA, which we are calling the messenger RNA. So mRNA. Then we've got what we call the tRNA, or the transfer RNA. Then thirdly, we've got what we call the ribosomal RNA. Okay, now these 3 types of RNA. What is the function of the messenger? What is the function of the transfer RNA? What is the function of the ribosomal RNA? Okay, now the messenger itself, messenger RNA, even just the name, it's a messenger. It carries a certain message. Okay, so the message that you've gotten from your DNA, okay, now you want to translate it to proteins. Okay, so this RNA is the one that is responsible for that. So it carries the genetic code. Now we're going to talk about the genetic code in details. Okay, so the genetic code, now the genetic code will just be a set of those instructions that will direct what type of proteins you're going to form. Okay, so it is usually made up of what we call codons. Now a codon is just, a sequence of, let me say, nucleotides, basically 3 of them. Okay, so basically that's what the mRNA is going to do. Okay, then the transfer RNA, even the name just suggests something. It transfers. So this one is going to transfer amino acids. Okay, amino acids for polypeptide synthesis, because you want to be making this polypeptide. Okay, you are going to, as we start now talking about the whole process, you're going to understand what role this one plays. Okay, then you've got a ribosomal RNA. Okay, so this is that part of the ribosome now that makes the proteins. Okay, that part of the ribosome that makes the proteins. So you know that this one's is going to be made in the nucleolus, and that is very, very important. Now, just like DNA replication, transcription, translation also has 3 steps. Okay, these steps we're talking about what? All these processes we're talking about, you need to initiate the process. Okay, initiation, elongation. Now elongation, in some literature they're going to call it polymerization. Okay, then after you've elongated your chain, what are you going to do? You're going to terminate it, termination. Okay, so basically these are the the 3 processes. Now I must make mention that these 3 processes, they are happening in the cytoplasm. Okay, because we're talking about a prokaryotic cell. Okay, they are happening in the cytoplasm. Now RNA, is made in the nucleolus. Okay, and it's going to go in the cytoplasm for these processes to happen. Okay, so basically that is it. Now let's say this is our prokaryotic cell. Okay, this is, you know, our nucleus. But now this nucleus is not a true nucleus. Okay, so after that, after it's been made from the nucleolus, let's say it's going to go like that. Okay, it has been made like that. It has moved from where it was and it has gone in the cytoplasm. This would be the mRNA. Okay, now the mRNA will be moving from 5 prime to 3 prime, like we saw in DNA, in RNA transcription. Okay, so that is the direction of the mRNA. You have to know that one. That is very important. Okay, now I thought of mentioning that before I talk about now the actual process. Let's start with initiation. To initiate is just to start. Okay, now what happens in is initiation. So we're talking about this mRNA running from 5 prime to 3 prime. Okay, so during initiation, mRNA is going to locate the ribosomal subunit. That is very, very, you know, as an initial step, mRNA locates the ribosomal subunit. Okay, now it's not just going to locate this ribosomal, you know, subunit. What is going to happen is that it can't just do that on its own, because that's when the process is happening. It's going to need some aid, okay, from a protein called an initiation factor. Okay, initiation factor. Okay, it's going to come, you know, to the aid. Okay, now this is my 5 prime going like that. This is to 3 prime like that. So this is our mRNA. Now as it is, you know, locating the ribosomal subunit, I must make mention something here. I don't know which one finds who, if it's either the mRNA or the ribosomal subunit, but they are going to meet, and when they meet, binding is going to take place. They are going to bind. Okay, let me say this is my ribosomal subunit. Okay, that is my ribosomal subunit there, and it's going to bind there. The process of initiation has started. Okay, so basically this is let me say this is a small ribosomal subunit. Okay, the small unit. Now basically it's going to be going in this direction as well. Now, so even the small ribosomal subunit is also moving in the 5 prime to 3 prime direction. Now what is going to be happening is that as it is moving in that direction, when it meets a 3 nucleotide AUG, let's say it meets it here, AUG. AUG is what we're calling a codon. A codon is triplet. It's got 3 nucleotides. Now if you don't really understand about nucleotides, you can also watch our video on nucleic acids. You're going to understand these nucleotides better and better, trust me. So we're talking about AUG. Now what is AUG? Let me just take you to the genetic code. Okay, this is the genetic code. So basically what is AUG? So the way you read the genetic code, okay, so I'm tempted to use the genetic code. So we're talking about A. So you start first with the first, even just the diagram itself, you know, it guides you on what you're supposed to do. So they're talking about the first base in codon, the second base in codon, and the third base in codon. So let's start with A. This is our A there. So A, then U is there, then where is G? G is here. Now where they meet is, what you want. So in this case you have A in this region, U in there, and G is here. So if G is there, so you need this one here, which is there. That is methionine. Okay, so basically that's it. Now on the other diagram, there are different genetic code diagrams. Okay, so even on the other diagram, that's what you're going to do. You get your A, so this is your A. You get your U is there. You get your G is here. Okay, so basically this diagram is a bit wrong, I think. Okay, yeah, it's wrong. So we're starting from here. This is 1, this is 2, this is 3. Okay, that's how you're going to go. Now you're able to see that AUG is here. Okay, and it's just showing us that AUG here is methionine and it is STAT. Even the name itself suggests STAT. Methionine is where you start from counting. Okay, once you locate the AUG, once the AUG has been located, it means now you are starting. Okay, now when AUG is located, here's what happens. Now a signal be sent to tRNA. So a signal be sent to tRNA to find an amino acid. Okay, so that is so once a signal has been sent to this tRNA, the tRNA now is going to bring the amino acid that codes for that. Okay, so this small ribosomal subunit is going in that direction. It finds AUG. A signal is going to be sent to tRNA to bring an amino acid that codes for AUG, but now an anticodon. An anti is the opposite. Okay, so that is what is going to happen. Now the start codon is different for different scenarios. Okay, if you are going to talk about eukaryotic cells and prokaryotic cells, so I'm always trying to differentiate the two because it depends where you're viewing this thing from. Okay, so for prokaryotic cells, the start codon is methionine, like we just saw. Methionine or AUG. Now in prokaryotes, it's not called methionine, it's called F-methionine, or other texts call it formyl. Okay, formyl methionine. So F-methionine. Notice the difference. Why I've mentioned this is because I've seen such kind of questions in the past papers. So you have to know this difference, and that is very, very important for us to understand. Now we've talked about the tRNA. How is the tRNA itself? Okay, now if I'm going to draw the tRNA just to make us understand, the tRNA has got arms, something that looks interesting. Okay, so if this is okay, trying to be artist. Okay, if that is our tRNA like that, it looks like this. Okay, so you're going to have this arm is shorter than the other arm. So basically the longer arm, it has a 3' end, the shorter arm has a 5' end. So this is how the tRNA looks like. Now the 3' end is called the acceptor arm. This one is called the acceptor arm. Now that is where the amino acid is going to attach here. Okay, now there's what is called tRNA charging, and there's also what we call amino acid activation. Now those small processes are very, very important because they're also examinable. So basically you have to also understand that. Now on the 3' end, you already have a certain codon. this you also have to know, is called ACC. Okay, ACC. So basically that's how your your tRNA is going to be moving from 3' end to 5' end, but in the middle you've got ACC. Okay, like that. And an amino acid is going to it attaches on the acceptor arm, the 3' end, and that is very, very important for us to understand. Now what is going to be happening is like this. Let's say, let me just reduce this diagram a bit to make it interesting. Okay, let me say it bulges like that. Okay, then this is our mRNA coming from the transcription that we just talked about, after it has undergone all the post transcriptional processes like that. So this is our mRNA. Now the ribosomal small cell ribosomal unit is going in that direction, and it has found the AUG. Okay, so let's say this is our AUG. Okay, once it meets that, signal has been sent to the tRNA to bring the amino acid that codes for that. So this that we have on the tRNA is called the anticodon. This that we have on the mRNA is called the codon. So this is a codon. On the tRNA you are going to have the anticodon. Anti to mean the opposite. Okay, so the opposite of AUG, that's what is going to be brought by the tRNA. So in this case, you know that A pairs with U will pair with A, G is going to pair with C. Okay, basically if you still don't understand this, I've emphasized you can also watch the video on nucleic acids. So there's base pairing, how we pair these bases. Okay, so that is very, very important for us to understand. So that is what is going to happen. Okay, this is the process of initiation. Now that, how does now this whole thing now elongates? Okay, how is it now going to elongate? So something very interesting is going to happen. Okay, so okay, I think yeah, so this is elongation. How does it grow now to become that big protein? Okay, so allow me to write my mRNA in a very straight chain, just for illustration's sake. So let's say this is our mRNA going to 3' end like that. Now you're going to have a large subunit come, okay, and you have a smaller subunit down. That's where the ribosome now comes in. Why? Because you already have the smaller subunit that is going in that direction, in the direction of the mRNA. So if that is our small ribosomal, this is our small, this one is our large ribosomal unit, and you know that the small and plus the large ribosomal, and that's what makes the ribosome. Okay, so this is a complete illustration of the ribosome, and that's what we said, the ribosome is the one that is involved in the making of proteins. But now how does this happen? So what we have here now is, okay, allow me to wrap that. Okay, what we have now here is so the ribosome is going to have 3 sites. Okay, this is what we are calling the ribosome, all of it, the small and the large subunit. So now the ribosome is going to have 3 sites. So let me call this one as E, this is going to be P, this is going to be A, EPA. Okay, now the E in this case is called, it means the exit. That's where your amino acids that are being made are going to be exiting from. okay, because you're going to realize that this is the mRNA. It has a lot of, you know, amino acids that are there. Let me say nucleotides. Okay, so 3 are going to be exiting, the other ones are going to be coming, and you're going to see that the P in this case, this one is called the peptide transferase. Okay, that is very important for us to understand. Then there is the A in this case. The A in this case is called the amino acid tRNA synthetase. Okay, synthetase. Okay, so that is very important for us to understand. Now, here is where now it gets interesting. Now, let's say we've found our AUG already, our start codon. Now, our start codon is going to be, let's say this is our P. Now, we started in P. I'm going to explain something very, very, important. Okay, initially, because that's when you're starting, so you're going to find your AUG in P. Okay, so now what is going to happen is that you found your AUG. Okay, your tRNA has come. This is our tRNA. Okay, it has brought the anticodon that codes for AUG, the opposite. Okay, and let's say this is the amino acid. It has brought that amino acid, that is, let's say, the 3' end, or which is the acceptor arm of the tRNA. It has come like that. Now we still need to move a step further. Okay, let's say, because you're going to have nucleotides here, 3 nucleotides, you're going to have 3 nucleotides here and 3 nucleotides there. Okay, now we found our AUG. We're going to have, let's say, at on the A site of the smaller subunit, or let me say the ribosome itself. Let's say you have UCC. Now UCC, if it's on A, what is going to happen again is that a signal is going to be sent again to, because you found your AUG, you've signaled, oh, we found AUG, we are starting now to code. So a signal is going to be sent to tRNA. It brings the amino acid that codes for that. Now we're talking about the anticodon, the opposite of AUG. That is what we have. Now we need to move a step further. A step further is A. Now on A you have UCC. So again you're going to signal to tRNA like, okay, we found another codon, UCC. So you need to bring the one that codes for that. So tRNA again is going to bring another, anticodon that codes for UCC. Okay, it brings, let's say this is our amino acid. Okay, that has happened. Now the anticodon for UCC, what is it? So let's go search for where UCC is on the genetic code. So we are talking about UCC. Okay, UCC. So okay, let me just use a different color. So this is U and this is C, and 2 C's of that. You're going to have 3 types of serines there. Okay, 3 types of serines. So let's also use the other diagram. Okay, so we're saying you get U, you get C, and another C. So now look at the times of serines that we have. We've got UCU, it's giving serine. UCC is giving serine. UCA is giving serine. UCG is giving serine. Now, what are you able to notice here? The UC is repeating itself, but however, the last nucleotide, okay, the last base is changing. Okay, so now our interest want UCC, which is here, but all of them are coding for serine. Now, this now brings the aspect of what we call the Wobo hypothesis. Wobo, or the Wobo hypothesis. So sometimes in a genetic code, you're going to tolerate the third, the third base. Okay, so that's what we're talking about. The first two, they should always be the same, but the last can, Wobo like that. It can change. Okay, but it is too code for the same. One of them is serine, and you're able to see most of these ones. Even proline here, you've got CCC throughout, but only the last one wobbles. Okay, so basically those are, even here 309, alanine, all those. So the third position has that kind of an arrangement. Okay, so that is very important for us to know. Now we found UCC. UCC is coding for serine. Okay, so an amino acid is going to be brought. Okay, that codes for serine, and it will come and bind there for the okay, that is our UCC there. And now what is now going to happen is an amino acid has been brought. You know that UCC in this case would be what? AGG, right? That is what you're going to have on the tRNA, okay, as an anticodon. Now that you have that, an enzyme is going to connect these two amino acids. Okay, that enzyme is what we are calling peptidyltransferase. Now, how does it connect to these ones? Because these ones are amino acids. What joins two amino acids? We know that what joins two amino acids are peptide bonds. So the peptidyltransferase is going to join, okay, these ones by formation of peptide bonds. Okay, the name the enzyme was saying is peptidyltransferase. Okay, now that is what is going to happen. Let's say the bond has been formed between these two amino acids like that. Okay, that is our bond. Now what is now going to happen is very, very interesting. Let me just go on the next page. Okay, this is the 5' end. This, okay, let me just write a bit down. Let's say this is our 5' end. This is our mRNA, 3' end like that. Okay, this is the exit, the peptidyltransferase, and the A site. Okay, now you have your small ribosomal subunit there. It's bound on the mRNA. Then you have your large ribosomal subunit, which is there like that. To make it complete, you found that this is the first one, the AUG on the P, which is there. Now this is the second one, the one just from coding, the UCC. Okay, now what is going to happen is that here we had AUG, okay, here we had UCC. So what is going to happen is that since you've connected the bond between the two, so what is going to happen is that the AUG is going to be on top. This is the AUG, then the UCC down. Okay, that's how they are joining. So let me just make it nicely here. So this is the UCC, this is the AUG, AUG on top, UCC down. they've been combined. That is the growing chain. Now look at where the growing chain is. It is in the A site. Okay, now what is now going to happen is that then after that has been made, after these ones have joined, the ribosomes moves 3 nucleotides in the 5' to 3' direction. Okay, 5' to 3' direction. So that is what is going to happen. So it's going to move here, this one is going to move here. Mind you, there's the E site here. Okay, then what is here also is going to move there. This side is going to exit. Okay, so that is what is going to be happening. So how does it happen? Okay, so when that moves, the AUG, when this one moves, let me just rub this part. This has moved from where it is here. It has occupied the second place. Let's say it has occupied this place. Okay, there and there. Okay, so don't forget, this is the first amino acid AUG, this is the second UCC. Okay, when it moves like that, the one that was the nucleotides which were on E, they are going to exit. Okay, exit like that. That's why, that's where the E is now. Now what do you notice? A is empty now. There is nothing in A. Okay, there is nothing in A. Okay, now in this case, bonds have been made and all those kind of things. Let's say now, if now we have at A, since it's empty, now let's say at A now we find we have now G, let's say GUG. If we have GUG on A, okay, let's go on the genetic code. GUG. So G U, okay, and another G. Okay, where is another G? GUG. It's here. Okay, let's also use the other one. We've got G U and another G. So that is what we are having. So where are they meeting? So we're finding that GUG is what? Valine. Okay, or valine. Now if that is what we have, a signal is going to be sent to tRNA to bring this, codon, and another let me say another codon is going to be brought by the tRNA. It's brought like that, and it's like that. So how does it join? Mind you, this is AUG, this is UCC, this is the GUG. So peptide transferase is going to form a bond between the UCC and the GUG. So, and when the bond is formed, so what are we going to have as our growing chain? Let me just show on the P side. Okay, this is what we have. So your GUG is going to come first, then you have your UCC, then you have your AUG, and another space again is because you've coded for this one. An empty space is going to be created there, and the chain will continue to grow. Okay, the chain will continue to grow. This is the process of elongation. Now I must mention something very important here. The only amino acids that come directly into the P site is coded by the AUG. Okay, That is very, very important. Otherwise, that's where most of the people tend to miss it. The only amino acid that comes direct into the P site is coded by the methionine or AUG. The rest go into the A site. Have you seen? The rest are coming to this one. It's empty and it comes just like that. The only one that came directly here was the AUG. All of them are coming here in the A site, and that is very important. Okay, now for tRNA to move from one site to another, let's say this is the tRNA. For it to move to the P site, from the A site to the P site, from the P site to the E site, that tRNA, for it to move, it needs an enzyme we call, because it is moving, so it is called, this enzyme is called translocase. Okay, for tRNA to move. So that is very important for us also to mention. Okay, now let's talk about the last part, termination. How does it terminate now, this growing chain that we're having? Now, just to draw a diagram again, we have a small ribosomal subunit there. Okay, we have our large ribosomal subunit. This is from 5' to 3'. That is our mRNA. We have the E site, the P site, and the A site. Okay, then let's say, you know, we have this is our growing chain there. Okay, that is our growing chain. It's growing and it's elongating. It's a big chain. We are adding amino acids. We are adding those codons, and it's becoming long and long. Now there's always a time to just terminate. If you've made more hair and more hair, there's just a time to stop. So basically, let's say it's going to break here. If you're breaking it like that and you want that part to be removed, initially we had what we call the initiation factor. Now, once, because you're saying the first acid, the first codon was AUG and it was in the P site. All of them that are now coming there in the A site. In the A site now, when you meet what we call the release factor, this is now what is going to signal. The release factor is what is now going to signal termination. Okay, so how is that going to be? Is when you meet a stop codon. Okay, so there are 3 stop codons that you're supposed to know. There's UAA. Okay, you have UGA and you have UAG. Okay, so basically these ones you are supposed to know. Okay, so these are known as stop codons. Okay, so like we've said, if you read any of these on the A site or the stop codons, a protein called release factor is going to come and bind to that A site. Then termination is going to happen. Okay, that is very important for us to understand. Basically, that's what happens, and it's going to be cut like that, and it has, it has been released. However, it's not active. It needs to be just the way we had made the mRNA. It underwent post-transcriptional modifications. Basically, that's what is also going to happen to these proteins for them to be active. Okay, but before I talk about that, let's talk about tRNA charging. How does tRNA charge? Okay, so now okay, so tRNA charging is something that is, let me just write it here. Okay, let me start first with amino acid activation. I think that's how it just makes sense. Okay, so let's say you have an enzyme. Okay, this is our enzyme. It has, you know, those things, an active site like that. Now we're talking about, because this one's mostly they happen simultaneously. Okay, this one happens and the other one follows. Okay, let's say that is an amino acid, it has bound on the enzyme. Okay, then let's say this is now our tRNA there and it also binds there. Okay, so I'm saying this is the amino acid, this is the tRNA. Okay, so now the enzyme that is going to be involved here is the, you know, we talked about the A site, right? Amino acid tRNA synthetase. The other one was synthase, but this one is synthetase. Now when we say synthetase and synthase, okay, it depends, you know, what you are meaning, because I was trying to check what is the difference between synthetase and synthase. Okay, so let me say synthetase and synthase. Okay, and this one we are saying it is the amino, you know, amino acid tRNA synthetase. Amino acid tRNA synthetase. Okay, synthetase. So basically this is the enzyme. Now synthetase, in this case, it means it is using energy. Okay, a very interesting process. So because it is using energy, this energy is in form of ATP. Synthase, on the other hand, it means it doesn't use energy, but in this case, this, because for it to be activated, it needs that energy. That's the difference. So now what are we getting to? What you are getting to is that the amino acid is going to be activated. Now how does an amino acid get activated? You know, this is where now the you know, attaching the amino acids to ATP, okay, and attached to the enzyme. When the amino acid is attached to this enzyme, so when the amino acid, this is the amino acid, okay, when it attaches to the enzyme, ATP is going to be connected, or it's going to be converted to ADP. This is ATP. So once the amino acid just attaches to the, you know, to the enzyme, in this case, your ATP is going to be converted to ADP. Why? Because you would have used part of the phosphate, okay, for energy. That is what we're calling amino acid activation. Now when tRNA, on the other hand, attaches to the enzyme, the ADP, because that's the energy that is there now, the ADP is going to be released. Okay, and once that happens, the amino acid now is going to attach to tRNA. Let me say they have attached like that. Okay, now this process of the amino acid attaching to the tRNA is what we are calling tRNA charging. Okay, so that's what happens. tRNA charging, it is now charged. Now the amino acid can now be transferred to where it needs to go, especially when a signal has been given to tRNA, bring such kind of an amino acid. This tRNA that transfers, that brings that amino acid to be coded for, or the anticodon, it has to be charged. So this is the charging process. It just doesn't bring, no, because the amino acid has to first attach, okay, attach somewhere so that it is taken now nicely. Okay, so basically that's what happens, but it's a process that requires energy in form of ATP. Okay, it's an energy kind of a process. So basically that is very important for us to understand. Okay, now we said now we've made our protein through that longest chain and it has been terminated. Now after termination, that protein has to undergo post translation. Post always means after, right? Okay, so post translation or modifications are going to now come in. Now there are a lot of things that happen based on what you want. Okay, so there are a lot. So a lot can happen, like methylation. Now methylation, it just means you are adding methyls to this compound, okay, to this inactive protein. You can also it can also undergo phosphorylation. Now phosphorylation, you're just adding phosphates to this inactive protein. It can also undergo glycosylation. Now glycosylation, you're just adding sugar molecules, sugar moieties, glucose, okay, to this compound. Okay, it depends with what you want. It can also be proteolysis. Okay, proteolysis, certain enzymes are made in an inactive form. Let's say this is our chain that has been made, our protein. Okay, now even in this protein that we've made, other proteins on this same compound, they are going to be inactive. Okay, that's why we just want the active part. Okay, that's why this thing is undergoing post translational modification. We don't want these parts. Okay, these parts they need to be cut. Now those parts that are inactive on this compound, okay, the inactive proteins are called zymogenes. So zymogenes need to be cut, and by what? Proteolysis. Okay, so when you cut those inactive parts, meaning you're step by step you're getting closer to something that is very active. Okay, so that is very, very important for us to also know. Okay, so you're going to cut some of the bonds to remove what is inactive and in order to activate our compound. So that is very important. And then other processes that can take place, what can we talk about? We can also talk about what we call N-acetylation. Now N-acetylation, you're just adding acetyl groups. Okay, you're adding these groups to the amino acid end of the amino acid, because what you should understand is that N, the way the amino acid is, it has the amino group, the NH, and the carboxylic end. Okay, amino acid, even the name amino because of the amine group, and acid because of the carboxylic acid. So the amino is the one that has N. Okay, it has nitrogen there, so you're adding acetyl groups to that N part of the amino acid. Okay, that can be part of the modifications. Then you can also maybe talk about lipidation, meaning you're adding lipids component or lipid components to this molecule so that it becomes a proper protein. So basically these are the steps on how translation happens, and that is it. So thank you for watching to the end. See you in other sessions where we're going to be talking about genetics.